Dear students, we have in the previous video seen that there were three factors and three-way analysis of variance, or with two interaction, or three factors we are dealing with at a time for all these 8 dependent variables. How we express these dependent variables, we have discussed that in the previous video. Now again, whenever you have a data like this, first of all, statistics, summary statistics, and descriptive statistics. From this descriptive statistics, you will see in the field, that is one factor. The change in the levels of the field, for example, you have 3 levels in the field, uh, how the mean, how the minimum, maximum 5-digit summary changes in these 3 levels of the field with respect to flag leaf area. So quartiles, minimum, maximum, coefficient of variation, median, we are opting all these statistics to report in flag leaf area with respect to field. You can also have all these dependent variables and it will give you a report collectively. We can select simply from this to this. Whenever you select this, press the shift key and then press this. This one, shift key and left click. Okay. Now press this. All these are selected and these variables will be broken down with respect to descriptive statistics of number of observations, mean, standard deviation, and you will choose standard deviation or standard error. One of this you will choose. Okay, CV, median, minimum, maximum, quartile. Okay. Now, temperature, and we will split it into field. So all these results will be splitted with respect to the levels in the field because field is our categorical variable. So if you have 3 levels, if you have 4 levels, mean, median, mode, all the descriptive parameters will be shown for each level of this field. Now see, here is F1, field 1, flag leaf area, tillers, plant height, this one, all these 8 variables. We can see, for example, 132 are the total observations. Okay. Mean of these 132 observation, flag leaf area mean is 21.29, standard deviation 9.1, CV is 42, quite higher. Minimum value of flag leaf area is 1.9 in field 1. Keep in mind, we are discussing field 1. That is in field 2. Then there will be in field 3. So flag leaf area mean of 132 observations in field 2 is 21.16 and here is almost same 21.29. So this is about every variable at a time with respect to the field. Okay, if you want to see a single variable at a time with respect to the field, how does it go? Let's see. Now we have flag leaf area in field. This is our variable F1, F2, F3, the simple one. Okay, as you will move in the same way for broken, this will be the same for this mean, standard deviation. You can also opt standard error. You can also opt minimum, maximum in the cross tab format. Okay, now see, this is the thing you will express in your paper or in your thesis. For example, flag leaf area with respect to field 1, field 2, field 3. Number 132 means, these are the means, collective means of field 1, field 2, and field 3. Standard deviation, minimum and maximum. Simple copy this and paste it in the Excel file and then convert that data table, text to table, and present in your thesis pages. And that will be presented like as I have shown you the MS Word file. Let me share that MS Word file here. Is it observable for you? Hopefully. Let's see this one. For example, this is our field or treatment 1, 2, mean for each, standard error for each, minimum, maximum, quartile, median, and differences. So when you move for analysis of variance, we have observed that कि जब आप एनालिसिस ऑफ वेरिएंस कर लें और उसका तरीकाकार जो है उसके बाद इसकी पोस्ट हॉक टेक्निक कैसे काम करती है. नाउ सी स्टैटिस्टिक्स, linear models, analysis of variance, this one. Again, write this route, statistics to linear model to analysis of variance to general analysis of variance. Okay. All these variables with respect to genotype, treatment, and reps. For field, you will exclude it only for field, only for reps. You cannot include this field with this treatment because the treatment is different in the field third. So we cannot include these two terms together. For the time being, that is genotype, treatment, and reps. First of all, you will select the term reps in your model, then genotype, treatment, then their interaction. Genotype with treatment. Okay. Now this is factorial analysis to some extent, not perfectly. For all these 8 dependent variables, click OK. Now this is analysis of variance. Replicates are not different. The p-value is above 0.05. Genotype shows significant effect on flag leaf area. Flag leaf area is significantly different in treatments, but not so much significantly different with their interaction. In this ANOVA table, we have interactive terms. So all this you can save. Again, this CV is higher for this flag leaf area, not so much validated. This one and all these variables at a time. You can copy them, paste them in Excel and make a single comprehensive table only with this mean square column and with this p-value for each variable. So all these 8 tables will be converted into one table to express in thesis or research paper. And I have shown you in the previous video how will you do that. Now, our post hoc technique. Go to the results. This is the first step. Our first step was to load the data in the statistics software. Second was to see the descriptive statistics. Third was to analyze with respect to the analysis of variance giving the model. And the fourth step now, we will go each step in detail further. Now, results. You have done analysis of variance, okay. Multiple comparisons, all pairwise comparison. Click that is the post hoc, okay. We are interested in treatments. We are not interested in genotype because we have 11 genotypes. We are not interested in one by one. We are just interested in treatments. How these treatments affect our all the variables significantly. And we will choose Taki HSD. Because most of the people choose LSD, that is for when you have controlled comparisons. For example, you are going to compare everyone with only control, you will go for LSD. But when you are going for treatments and every treatment is being compared with every treatment, so that is multiple pairwise comparison. So in this pairwise comparison, when you are going T1 to T1 is going to compare with T2, then T3, then T4, and every treatment is going to compare with everyone. You will recommended to choose. You are recommended to choose Taki HSD, not LSD. So we will go with Taki HSD, and HSD mean honest significance difference for treatment. Okay, for example, if you go with these three, these are unnecessary, but we can also go with these. Now, okay. And this is done for 8 variables at a time. Look how long this file is. We do not need this all. So this is the reason I was saying high level of comparisons we don't need, because we have 11 genotypes. We need only this one. We are interested in treatments. We are not interested in any other comparison. Or so we see statistics, linear models, analysis of variance. This one, this model is always there. You can have you can note it further. Okay, then again post hoc, for post hoc results, multiple comparison, all pairwise comparison. Remove these terms. We can have genotypes. Okay. Okay. Now see, the results critical value is variable varied for due to this and this. See, these are the classes in our treatments. T2 is significantly different from G4, significantly different from T1 with respect to flag leaf area, with respect to temperature, sorry, with respect to pillars, with respect to any other like plant height, we can see the similar letter mean these 2, 3, 4 averages are not significantly different from each other. But this one is different from this, and this one is significantly different from this. So there are 4 groups, A and B, in which the means are not significantly different from each other. You have to copy these results. If you go multiple comparison, all pairwise, just remove this, go with this. You have only now treatments. Just copy and paste the results, and these 3 columns will help you to give a bar chart. Bar chart with, for example, bar chart with letters. You can also make bar chart here. Go with plots, plots with the means. You need means for treatments, mean factor, or place genotypes in mean, or treatments in bifactor. We need bar plots. We can also have line charts. Click equal. The number of levels of vector treat exceed the maximum number of 6. Okay. Go with genotypes only for the timing. It cannot. We have 11 different treatments, 11 genotypes. So our treatments are much higher, 3, and these are also count that 1, 2, 3, 4, 5, 6, 7, 8. 8 treatments are there. So it only takes 6 treatments. For the time being, that is the genotypes. Behavior for flag leaf. You can just simply click this. Almost TMP is the same, not significantly different among genotypes. Tailors are same, not significantly different among genotypes. You can set this graph as in like published data. We are setting its size. Now, this is the size. You can simple copy this and paste anywhere. Very easy to understand. Okay, the next is plant PL, peduncle length maybe. So peduncle length only goes for 60 for each variety. You can also have may generate the graph for treatments as well. So this is the graph generation from statistics.